Happy Monday. Hopefully you guys had a good weekend celebrating Father's Day or something like that, just kicking back. Um, yeah, my weekend was good. I went up to see my folks. My dad's 90, so it was really nice seeing my both my parents and in the midst of all this COVID stuff, it's been pretty challenging. So, but do what we got to do, right? All right. So we've got 2 weeks left. Actually, yeah, a little less than 2 weeks. So let me kind of show you what our plan is for this week. Today we're going to cover the second renal lecture. The first one was kind of more of an osmoregulation. If you remember, we kind of focused on how different animals deal with regulating water and salt, different vertebrates, and then we ended up really talking about the vertebrates as far as mammals, where their main organ for osmoregulation is the kidney, whereas obviously other animals like amphibians have their skin, fish have their gills. We talked about all sorts of different mechanisms. So we started talking about kidneys, and between today and tomorrow, we're going to really just get into the anatomy and physiology of the kidney. I'm kind of breaking away a bit from just animal physiology in general, and I'm really going to focus just on the kidney, really the same information that I would give my human physiology class. So, which hopefully will be helpful for you. We'll do that today as well as tomorrow. Both lectures are up. And then on Wednesday and Thursday, we're going to start with the endocrine system, one of my favorite systems. I haven't posted those lectures yet, but they will hopefully be up by sometime tomorrow. And we'll do that. And then next week, we're going to wrap things up. I'm not sure if we're going to quite make it, if you look at the syllabus, to thermoregulation. I hope we do, because it's really interesting, but we'll kind of see how things go. However far we get, our last day of class is July 2nd, when we'll have the exam. So maybe July 1st will just be really a review. All right. So moving along here, let's see, new share. All right. So like I said, today we're going to really today and tomorrow we're going to focus on more details, more of the anatomy, physiology of the kidneys, talk a little bit more about what they do just besides regulating salt and water. All right. So when we talk about the urinary system in general, and again, I'm really focusing here on, primarily the focus is on mammals. There are 4 main organs of the urinary system. The kidneys, which are really the workhorse of the urinary system, these are the organs that actually filter blood, modify it, and ultimately come up with an excretory product that gets eliminated, kind of processing the filtrate. The remaining organs, not any less important, do different things. The ureter, or the ureters, are the tubes that carry the newly formed urine from the kidneys into the bladder. So these are the 2 ureters. They transport urine to the bladder, and of course, it's the bladder where the urine is stored until urination occurs. And when the signal for urination, which we'll talk about next time, the mechanism for it, neurological and things like that, when that occurs, the urine is actually expelled from the bladder, goes through the urethra, and to the exterior of the body. So really the primary focus, I'd say about 95% of these next 2 lectures are really going to focus on the kidney. All right, so what are the functions of the kidneys? So we've been talking a lot about osmoregulation, right? Regulating salt-water balance. in different animals. And certainly that's very important. That's a very important role for the kidney in mammals. In addition to regulating osmolarity, and kind of in conjunction of it, by regulating the salt and water balance, that has a secondary effect of regulating the amount of fluid in the body, as well as blood pressure. So even though the kidneys we often think of as an excretory organ, that really is not doing it justice, because it's more of a regulatory organ, regulating the composition of blood, which ultimately results in waste products being eliminated, but other substances being returned to the blood. And finally, there's another role on regulating pH. We all know that what the physiological pH should be. And the ability to regulate pH is really determined by the respiratory system in one aspect. We talk about CO2, right, carbonic acid, but also the kidneys play a very important role in regulating pH. Once all this regulation is complete, whatever is not conserved or returned to the blood is now destined to be eliminated. Excess water, metabolic waste, as well as toxins, things that can build up inside our body from medication and other substances that we need to get rid of. One other function of kidneys that we don't often talk about as much, but I think we already have spoken about it in a different system, is the production of hormones by the kidneys. One hormone that the kidney makes is known as erythropoietin. We spoke about that when we're talking about how erythropoietin is important when oxygen levels get low, which can be due to anemia. Erythropoietin stimulates the production of red blood cells and hemoglobin. So you can see kind of the interrelationship between the importance of kidneys and a lot of cardiovascular functions. And that's why often when you hear about someone who's suffering kidney failure, it affects cardiovascular. Or if they're suffering cardiovascular problems, it affects the kidneys. All right. Let's take a look at some anatomy, because I think it's really important to go over the anatomy, because that will give you an appreciation as far as everything that's taking place in the kidney. If you look at the kidney, you will see a couple millions and millions of a couple different components. One we call the renal corpuscle. The renal corpuscle, and there's millions of these, consists of a set of capillaries known as the glomerulus, which is what this is right here, and a tissue that surrounds it called the glomerular capsule. So what I'm encircling right now is the renal corpuscle. We're going to talk a little bit about what's going on here. I'm just kind of identifying the parts, the structures. So you have the renal corpuscle with the capsule surrounding the capillaries. And then if you follow the yellowish color, we have all these different tubes that are going through here, okay? First set of tubes I want to mention are called the proximal convoluted tubules. This connects with the structure you've heard of already, the loop of Henle. This is where a lot of concentrating of urine takes place. And there's a couple different parts to it. There's what's called the descending limb, the ascending limb, but I'm not going to ask you those details. Just know the loop of Henle. So from the proximal tubule, we reach the loop of Henle. From the loop of Henle, we reach the distal convoluted tubule. And of course, you can see why they're called convoluted. They have this kind of coiled appearance. From the distal convoluted tubule, the urine then reaches what's called the collecting duct. All right, so there's a lot of information here, but just to kind of narrow it in scope, you can picture that the kidney is a major organ for filtering blood, right? So blood enters the kidney, is filtered across the glomerulus, and the fluid that filters across the glomerulus enters into these tubules. First, the proximal tubule, next the loop of Henle, next the distal tubule, and then finally the collecting duct where it eventually leaves the kidney. So you might wonder, what's the purpose of these tubes? Well, the purpose of these tubes is to modify the filtered blood. The material that filters from the blood into the kidneys, right? A lot of it has to be modified. A lot of what enters into the kidney goes back into the blood. A lot of other material gets secreted. So we're going to take a look at each of these different regions just to kind of have an idea of what's taking place in each area. But this is just kind of a general view of the anatomy. So we've got this renal corpuscle plus all these tubules. Collectively, that's known as the nephron, right? So here is our renal corpuscle. Here is our proximal tubule. Here's our loop of Henle. Here's our distal tubule, and here's the collecting duct. This whole thing is known as a nephron, and there's millions of these in each kidney. All right, some more interesting kind of key points about the kidney is, as I mentioned, this is where blood is filtered. And of course, how does blood enter into the kidney? Through what are called the renal arteries. And they enter the kidney through what's called the hilus, which I'll show you what that is in a little bit, but it's just a region where the urine enters, or the blood enters, I should say. Just to give you an idea of how significant the blood supply is to the kidney, about 20% of the cardiac output enters through the kidney. So about 1/5 of the cardiac output, so that's about 1 liter per minute, enters into the kidney. That's a lot, right? So it basically takes 5 passes for the entire blood to be filtered. That's a lot of blood being filtered in the kidney, considering the fact that the kidney makes up for less than 1% of the body weight. But because of that activity, because of all the different filtering going on, there's a lot of ATP being used. So a lot of energy being expended, and obviously a critical organ. Granted, some people have to have a kidney removed. They're able to get by with 1 kidney. Preferably, of course, if someone loses a kidney, there's a transplant available. But nonetheless, obviously, there's a lot going on in the kidney. A lot of blood passing through, a lot of energy being expended. And we're going to take a look at what that energy is being expended on. Okay. Function of the kidney initially is basically to filter blood, and then the renal veins exit. So let's take a look first of all where the hilus is. All right, this area right here is known as the hilus. I don't think there's a label on it, but you can see this is the renal artery going in. This is the renal vein leaving. Now you'll notice once the renal artery enters into the kidney, it splits off. And it splits off into several branches. I want you to know these branches, both the arterial and the venous. From the aorta, the renal artery splits into the segmental artery. You can see these right there, into what we call the interlobar arteries, into the arcuate arteries, into what are called the cortical radiate arteries, which are these small little ones up here. Finally, into the afferent arteriole, into the glomerulus, right? And that's where the blood is filtered. Now, when I say the blood is filtered, I don't mean the kidney fills up with the kidney when it's filtered, all the blood goes in. No, what goes in is really the plasma, the fluid portion of the blood. All the cells stay in the blood. They're not filtered into the kidney. What stays in the blood ends up entering into the efferent arteriole and passing through a series of venous vessels that carry it out. Now, one really cool thing about this is you'll notice that the veins and arteries, for the most part, have the same names. Renal artery, renal vein, interlobar. There's no segmental vein, of course. There's arcuate artery and vein, cortical radiate artery and vein. The difference, of course, is the peritubular capillaries. So what I want you to know is really the order of how the blood enters into the kidney and how the blood leaves the kidney. All right. So this is a close-up view showing some of the details more close up, and you can see, for example, closer to where we're looking at the glomerulus. So you remember from the renal artery, it splits off into the segmental, into the interlobular, eventually. And here, I want to mention this key point right here. This is what's called the afferent arteriole. It's the afferent arteriole that carries blood into the glomerulus. And that blood is going to be filtered or squeezed across the capsule into this proximal tubule. One way to think of it is to think of it as a piece of cheesecloth being filtered, filtering blood that way. So a lot of the fluid portion gets filtered across into here where it's processed, and we'll take a look at that. What is not processed, what does not leave the glomerulus, ends up staying, passes through the efferent arteriole, right? And you can see all these other vessels as well, all the different veins. These are the capillaries, and eventually exiting through the renal vein. Okay, so hopefully you've got an idea of the blood supply and the blood drainage of the kidney. Now let's get into really the physiology of the kidney. As I mentioned before, the kidney, of course, is the principal osmoregulatory organ of mammals. Remember, I mentioned many birds, of course, have salt glands. Not all of them do, but it's the principal osmoregulatory organ in mammals, because our skin is not really a major osmoregulatory organ. We might lose some salt through sweating, but it's not a major factor. And obviously, we don't have chloride cells or gills, and our skin's too thick. So anyway, so what takes place in the kidney? So as I mentioned before, the very first event that occurs at the kidney is blood entering through the afferent arteriole into the glomerulus is filtered or squeezed across into this structure, which is known as either, you can call it the glomerular capsule or Bowman's capsule. Bowman's capsule is the old school way of calling it that, but the new version is glomerular. So either is fine though. That's what's known as filtration. Now, once the filtered blood enters across the capsule, enters into what's known as the proximal convoluted tubules, okay, most of what is entered into the tubules returns back to the blood. This is what's called reabsorption. What types of things are returned back to the blood? A lot of water, most salts, glucose, most things that we really need, amino acids, stuff like that. Now, granted, obviously, when we do pee out water, and a lot of that has to do with the amount, right? If we have an excessive amount of water, we will pee out the excess, but a lot of it is reabsorbed or returned back to the blood. And you can see following this letter B, how it's returned back. A little bit further down in these capillaries, we see something also quite interesting, is the fact that this arrow looks like blood is going back from the capillaries, back from the blood, back into the kidney. It's like, what's going on here? This is what's called secretion. And secretion is where materials that were not filtered into the kidneys at the glomerulus enter into the kidneys through this process. Now, what's the difference? Here we see a lot of filtering, mostly of water, ions, minerals, sugar, things like that. With the secretion, this is where we see movement of toxins, metabolic waste. Maybe you took a Tylenol, right, for a headache. Well, that's great, the Tylenol helped, but when the Tylenol or the acetaminophen breaks down, there's particles in there, there's molecules that are not really good for you. So you need to excrete them. And they typically get excreted this way, through secretion. Once all these steps have been completed, the filtration, the reabsorption, and the secretion, the end result is we now have excretion, or the final composition of the urine. All right. So the urinary excretion of a substance depends on how much is filtered, how much is reabsorbed, how much is secreted, and that gives us our amount excreted. So there's a little equation right here. If you take the total amount that's filtered, subtract what's returning back to the blood, adding what's being secreted in, we come up with the total volume that's going to be excreted. Now, of course, that's going to vary from individual to individual. It's even going to vary within that individual, right? Depending on their diet, on their fluid intake, maybe their general health. All right. So just like other different body systems, you know, when we're looking at how well they're functioning, there are certain parameters, certain factors we study. For example, with the heart, we talk about cardiac output, right? With breathing, we sometimes talk about breathing rate or tidal volume, right? With the kidneys, we often talk about the excretion rate. And the excretion rate, of course, refers to the amount that's excreted, which is equal to the amount filtered plus the amount secreted minus the amount reabsorbed, which is exactly what we saw back here. Oops, exactly what we saw back there. Okay. So the amount excreted depends on three factors: how much we filtered, how much we secreted, and how much we reabsorbed. Now, if we're looking at a particular solute, and this can be studied, if you're looking at sodium, if you're looking at glucose, it's possible, it is possible to assess what's happening to that solute if you look at a couple things. If you find out that the amount of solute excreted per minute is less than the amount that was filtered initially, that means the solute was reabsorbed. In other words, if we have, say 10, I'll make this up, 10 milligrams coming in, but say 5 milligrams being excreted, that means some of that was reabsorbed. If, in contrast, if the amount that you excrete is greater than the amount was initially filtered, that means some more of it was added in, and that's where more is secreted. And there are certain substances that we know for a fact are reabsorbed. For example, glucose. Normally, we don't pee out any glucose. It's all reabsorbed. Obviously, that depends upon if somebody is a diabetic, if they're being treated for it, other factors. So just look at that relationship. If what's going out is greater than what's coming in, that means there was some secretion of that substance. If the amount leaving the kidneys is less than the amount that came in, that means it's being reabsorbed. So it's kind of intuitive. All right. All right, so just to kind of throw out some numbers for you, we can just take any substance. So let's just say a solute. The amount filtered was about 12 millimole, right? The amount secreted is 3. So 12 was filtered, we added another 3, but then we reabsorbed 6. So that ends up with an excretion rate of 9, right? So we had a net reabsorption, right? Because we came in with 12 and we left with 9. So more was reabsorbed than what was secreted, right? So that's kind of one way to look at it. Every solute is different, but that's just kind of an example how you can assess what happens to each solute, because some solutes aren't secreted, some are just reabsorbed. All right. So to kind of summarize, filtration involves the production of a filtrate, which is really the plasma, which leaves the glomerulus. Most of what enters from the glomerulus into the tubule is reabsorbed. Then that includes nutrients, water, essential ions. They're all returned to the blood. About And this, I'm not exaggerating, roughly about 99% of the solutes that enter into the kidney are reabsorbed. 99% of what goes into the kidney is reabsorbed. So, and that's really important as land animals, right? Especially even with the water, right? We've got to conserve the water as well. What typically is secreted, what I call undesirable molecules. That would be, again, toxins, metabolic waste that we don't want building up in the blood. All right, so where does filtration occur? All right, well, I already really told you that, but just to elaborate, filtration occurs at the renal corpuscle. All right, now this is a different view from what we saw before, but here is the afferent arteriole. This is the glomerulus. This is the capsule. This is the efferent arteriole. This is the proximal tubule. So blood enters through here, is squeezed across, and goes into the proximal tubule. Now, when we're talking about filtration, it's kind of a similar scenario when we're talking about respiration. Obviously a different system. Remember when we're talking about respiration, especially with the alveoli of lungs, I mentioned that it was very important to maximize respiration by having a very thin respiratory membrane, very flat cells, well vascularized, stuff like that. Well, with the filtration membrane, it's also quite thin, but it consists of 3 parts. So the filtration membrane are the layers that the plasma has to pass through to get into the Bowman's capsule. So it has to, once it enters into the capillaries, glomerulus, how many layers does it have to go through to get into here? First, it has to pass through the capillary endothelium, right? This is the simple squamous, these flat cells that have pores in them. Remember we spoke about fenestrated capillaries? This is a good example of where we have fenestrated capillaries. So what passes through the fenestrated capillaries crosses what's called the basement membrane or the basal lamina. Then it encounters another layer, and this layer is made up of portions of what are called podocytes. Podocytes are cells. They're very similar to what are known as astrocytes. These are star-shaped cells in the brain that are important in the blood-brain barrier. But if you can see this is a podocyte. Here's the nucleus. And you can see all these white extensions. It kind of wraps around the capillary. Well, there's these little spaces in between. And these spaces is where the blood is filtered. So the filtration membrane consists of the capillary endothelium, the basement membrane or the basal lamina, and then the podocytes. What gets across there is now what we call the filtrate, and that's what's going to eventually get into the proximal convoluted tubule. All right. So how do we, remember I mentioned, how we assess things, how do we assess how the kidneys working, how do we assess how lungs are working, heart's working? Well, I already mentioned the excretion rate, right, is one way to assess how the kidneys are working. But there's some other parameters where we can measure really how much is being filtered or how fast everything is being filtered. And this is what's called the GFR, otherwise known as the glomerular filtration rate. And this is the volume per unit time that the plasma, without any large molecules such as proteins, without any cells, just the water, the salts, the glucose, amino acids, how quickly that passes from the glomerulus into the capsule. And it's been determined that it amounts to about 125 milliliters per minute, or about 180 liters per day is filtered. Right, that's a lot, right? A liter is about a quart, right? So 180 quarts, what's that, like 45 gallons? It's a lot. And it has to obviously pass across three barriers, the filtration membrane, to enter Bowman's capsule. Now, if you think of that volume of plasma, filtrate, going into the kidneys, 180 liters per day, you certainly don't pee out 180 liters per day, right? If we did, we'd be in the bathroom all day long. We normally only pee out maybe about 1 liter or 1.5 liters a day. And that's because most, like I mentioned, most of what goes into the kidney returns back to the blood. All right. So GFR, glomerular filtration rate, is about 125 mils per minute, 180 liters per day, right? And of course, that's the rate at which that protein-free plasma is squeezed across the glomerulus into the proximal tubule, right? Well, as it turns out, there are certain factors that can influence it. Now, hopefully you remember when we were talking about capillaries in general, when we were talking about the cardiovascular system. I mentioned that with the arterial end and the venous end, at the arterial end, fluid leaves the capillaries, goes into the tissues, right, due to the relationship between hydrostatic and osmotic pressure. And at the venous end, right, fluid leaves the tissues, goes back into the capillaries, and this is due to, again, a change in the relationship between osmotic pressure and hydrostatic. Not change in relationship, but the change in the amount of pressure, okay? Well, something similar is found with the kidneys. However, in this case, there's 3 distinct pressures that influence how effective or how much GFR we're going to have. These are what we call the Starling forces. And there's 3 forces that determine what the GFR is. First one is what we call the glomerular hydrostatic pressure. And you could say that's just blood pressure. That's blood coming in through the afferent arteriole, right? And that's pushing, right? That has a positive drive. It's going in this direction. You take a look at these arrows, right, going this way. So that favors GFR, right? Well, then we actually have 2 opposing pressures. We have what we call the plasma osmotic pressure, sometimes called colloidal, which is due to the proteins that are in the glomerulus that are drawing fluid back this way. So that's opposing the outward flow. There's one more pressure that also opposes flow, and that's what's called Bowman's capsule hydrostatic pressure, or just capsular hydrostatic pressure. That's the fluid that builds up once the filtrate enters into the capsule, right? You build up fluid. That fluid is going to push back in this direction. So that's going to oppose it. So we have 1 pressure favoring it, 2 pressures opposed. And that doesn't sound like the odds are very good that we're going to have any GFR. But fortunately, we do. And that's because the hydrostatic pressure in the capillary, in the glomerulus, is high enough. And it's typically around 55 millimeters of mercury. That's the pressure that's pushing the blood through the glomerulus across the filtration membrane. Right, that's going to be this one. Right, that's this one right here. Right. The Bowman's capsule hydrostatic pressure is about 15. That's going to push back. We also have the glomerular osmotic pressure, right? And that's also going to pull blood back. That's due to the protein in the blood. So we've got 55 going one way. We've got 30 and 15 going the other. Fortunately, we still have a positive value, right? If we take 55, driving this way, oppose it by 30, oppose 15, we still end up with a positive net pressure of 10. That's all we need, as long as it's positive. If these 2 pressures exceeded this one, obviously that would be a serious problem, and your kidneys would fail. You wouldn't be filtering anything, and If that's not corrected, someone would die. Okay. So we need something like this to maintain the GFR of 125 mils per minute. All right. Okay, so just to kind of go over some of these terms again and add a few. So when we talk about the Starling forces or the filtration pressure, we've got, and these terms I haven't used before, the glomerular, this is the hydrostatic pressure of the glomerulus, that's 55. We subtract that from these two, and we end up with 10. That's the Bowman's capsule pressure, and that's the osmotic pressure. So we have a plus 10. Now I threw in this number, and I'll explain that in a little bit. There's another value that's called the renal plasma flow, and that's actually the amount of blood that's actually entering into the capillaries, into the glomerulus. There's a difference between the blood that's entering, which is 625, and the actual amount that's being filtered. If you take 125 over 625, that ends up being 20%. Oops. Remember I mentioned about 20% of kind of what goes in is filtered, right? So, or ends up going into the Bowman's capsule. All right, hold on one second now. All right, so just keep up that renal plasma flow on the top of your mind there. Okay, so let's take a look at some more numbers. So GFR is the amount of fluid that enters into Bowman's capsule, whereas 625, otherwise known as the renal blood flow, that's the amount that actually enters into the capillary but is not yet filtered. And most of it, of course, is not filtered. Only about 125 is. All right. This is where it gets kind of really interesting. You can kind of appreciate this. If 180 liters per day reaches Bowman's capsule, remember I mentioned a lot of what enters into the kidney ends up being reabsorbed? By the time that we get to the end of the proximal tubule, only 54 liters is left. By the time we get to the end of the loop of Henle, there's only 18. By the time we get to the end of the collecting duct, that's kind of like the final frontier, right? What you have there is what's going to be peed out. You've got 1.5 liters. So we went from 180 liters down to 1.5. That's a very powerful organ, right? That can actually effectively remove, or basically conserve, I should say, really conserve a lot of the essential components and ensure that they're not eliminated. And again, this is an average number, right? It's going to vary from person to person. All right. So we've got, if you take a look here, and just to kind of reorient you, this is meant to be Bowman, the glomerulus. This is the afferent arteriole. This is the efferent arteriole. This is the proximal tubule. And of course, filtration is occurring over here. Remember I mentioned about 99% of what enters into the kidney ends up being returned back to the blood? Well, there it is right there. Okay, so let's kind of go back some of the different numbers. So remember I mentioned about 625 mils per minute or renal blood flow enters into the glomerulus. Of that 625, only 125, which is 20%, enters into the proximal tubule, right? And of that 125, right, mils per minute, most of it ends up being returned back. That 20% number that I spoke about before is also known as the filtration fraction. So the filtration fraction is how much of the actual blood that enters into the glomerulus, how much of that is actually filtered. And at one time, it's 20%. So basically, it takes 5 rounds through the body for the entire blood to be filtered. Okay, so here we go again, just to kind of finalize everything. So filtration fraction is equal to GFR over the renal plasma flow. 625 mils of plasma enters each kidney or the kidneys per minute. 125 mils is filtered into Bowman's capsule per minute. 125 over 625 gives us a filtration fraction of 20%. And this is important. All these numbers are important, and these parameters can be assessed. And if they're off by any amount, it can indicate someone is starting to experience, or maybe early or possibly even late stages of kidney failure. And I know some of these are repetitive, but this is really kind of showing the same thing. All right, so 180 liters are filtered per day. Only 1.5 liters is excreted. That's less than 1%. So, and this is dependent upon, in order to have this system, we not only have to have good filtration, GFR, right? We've got to keep that up. And that's why blood pressure, we'll get into this later, but remember, hydrostatic pressure is really important for GFR, right? That's that 55 millimeters of mercury pressure going through. Say if someone's got low blood pressure, that low blood pressure translates into not as much blood pushing into the kidneys, and so that can lead to kidney failure. So your blood pressure, obviously it affects many things, but one of the things it can affect is the kidneys. The blood pressure is too low, it can affect the kidneys. The blood pressure is too high, it can affect the kidneys. All right. Now I mentioned that GFR is regulated. Now when I say it's regulated, I mean, we've already shown that there's 3 different pressures that can influence that GFR, right? We spoke about the forward pushing hydrostatic pressure, the capsular hydrostatic pressure the other way, and the osmotic pressure the other way. Well, we also have some other ways of ensuring that those pressures stay what they're supposed to, or interfering with some other factors. There's 2 main different mechanisms that can influence GFR, or basically help to keep it at the 125. One is called myogenic regulation. And this is an important, very important adaptation that works in most of us that responds to high blood pressure. So say, for example, if someone's blood pressure goes up, right? Like I mentioned before, we want that 125 mils per minute to pretty much stay constant going through the kidney. If the blood pressure is going to go up, that's going to increase the GFR. But we don't want a high GFR, we don't want a low GFR, because that's going to affect the function of the kidneys. So when the blood pressure goes up, those afferent arterioles, and if you remember, arterioles have a lot of musculature, right? Remember, we spoke about Puiseuil's law, right? We spoke about diameter and diverting blood, regulating blood flow. If the blood pressure goes up, those afferent arterioles will dilate, and then they'll constrict. And when they constrict, that will kind of make sure not an excessive amount of blood enters into the glomerulus, and thereby maintaining the appropriate, the physiological GFR. So that's what we call myogenic regulation. That's kind of a direct effect. The other one is what we call a tubuloglomerular feedback. And I've got some pictures to show this, because this writing doesn't really do it justice. There's these specialized groups of cells that are primarily found in the distal convoluted tubule. They're also found in the collecting duct. And what they do, they are kind of what I would refer to as chemoreceptors. We've spoken about baroreceptors. Remember, cells in the carotid artery and in the aorta that respond to changes in blood pressure. We've got these chemoreceptors that respond to levels of solutes that are passing through the kidney. And if they detect the levels, the fluid levels or the solute levels are too high, they secrete a substance, and I'll show you a picture of this, called adenosine, You've heard of adenosine triphosphate, right, ATP, but adenosine in this case actually has a different effect by itself. Adenosine is secreted and actually causes those same smooth muscles that we saw up here to contract as well. So really what's happening is these two things are both responding when the blood pressure or the blood volume gets too high. They both respond similar to try to reduce the flow to maintain GFR. They just do it a little bit differently. One acts directly, is where the arterioles respond directly. They stretch and then constrict. Whereas these, we have these maculodensa cells that secrete a substance that causes the arterioles to constrict when a certain level of solute is detected. And I'll show you some more details about that. All right. So let's take a closer look at the myogenic response. We sometimes call this an autoregulatory mechanism because it's auto meaning it's doing itself. You heard of like autocrine secretion, where a cell produces a substance that's affecting that same cell. This is a diagram that shows a couple things. Let's kind of orient ourselves. This is the afferent arteriole. This is the glomerulus. This is the capsule. And this red arrow is meant to show GFR. And note the size of that arrow, right? And this is the efferent arteriole. So take a mental snapshot of this picture. Blood's coming through here. Okay. If blood pressure gets too high, look what happens. See this? If we go back for a second, there is this part of the arteriole, it's normal size. There is a constriction that occurs that results in GFR decreasing. You notice with GFR, oops, remember GFR was like this? GFR is now lower. GFR decreases because it was increasing with the high blood pressure. This helps to ensure that GFR is maintained. All right. Now, this is actually really shows the extent and how incredibly effective our system is in regulating GFR. You know, I've already shown with the myogenic response, tubuloglomerular feedback, which is with the maculodensa cells. But just to give you an idea how effective it is, remember I mentioned that GFR is 125 mils per minute, or translated into 180 liters per day, right? And this is showing what the blood pressure is. Now, how do we translate this diagram? If you take a look at where this line is flat, this shows when the mean arterial pressure is 80 or even as high as 180, the GFR does not change. I mean, that's pretty amazing how through those mechanisms that I spoke about, through myogenic response and through tubuloglomerular feedback, our blood pressure could go up, but through those regulatory mechanisms, we keep the GFR at 180. Obviously, the system isn't perfect, and if the blood pressure is really high, right, we can't regulate it, and the GFR is going to go up, and that's going to cause kidney damage. Now, what about this? When the blood pressure drops really low, you notice GFR drops really low. Now, what's the purpose of this? This is actually a response to hemorrhaging. When the blood pressure drops like this, it's a pretty good indication that might be some internal bleeding. And so essentially what happens, the kidney shuts down as a way of ensuring that excess fluid is not lost. In other words, if you're hemorrhaging, why would you want to filter more blood through the kidney? So basically, it greatly shuts down GFR, thereby trying to conserve whatever blood volume we have. It may not be enough, the person might not make it, but it's an adaptation that occurs. Oops, what am I doing here? Okay. All right. So speaking of hemorrhage, this is kind of an interesting diagram, and it kind of goes in hand with when we're talking about low blood pressure. Remember, we were talking about low blood pressure, how that affects how the cardiovascular system responds by increasing sympathetic activity to increase cardiac output, right, and to increase resistance. Well, just take a look here. There it is right there. But at the kidneys, that same drop in blood pressure, in this case due to hemorrhaging, causes a decrease in GFR, a decrease in urine flow, and thereby decreasing fluid loss. So that's kind of going, so this right here is going to result in a decrease in fluid loss, trying to conserve whatever fluid we've got. All right. So, summary. Summary of the myogenic response is that an increase in blood pressure stretches the afferent arterial smooth muscle, that would be the tunica media, causing what's known as a vasodilation, right? So it's dilation means to enlarge. And then it's kind of like a rubber band, it stretches and then it recoils to constrict. This results in a decrease in blood flow and GFR from when it had shot up because of the blood pressure. Now it's bringing it down to a bit more of a physiological level. If blood pressure falls below 80, as you saw from the previous diagram, there's a decrease in GFR. There's no vasodilation. In fact, there's a vasoconstriction, which will conserve fluid volume. Right, so this is all involving what's happening at the afferent arterial. What about the efferent arterial? Okay, so efferent arterial, it's kind of an interesting area, but the effects of vasoconstriction have an opposite effect on GFR. So remember we were looking at the afferent arterial. When that constricts, it's going to reduce renal blood flow, thereby reducing GFR. When we constrict the efferent arterial, GFR goes up. Now, why is that? Well, if you think about it, we've got all this blood coming in, right? And there's nowhere for it to leave. The exit through the efferent arterial, remember what isn't filtered normally leaves? It doesn't leave, so it ends up being squeezed across here even more. Now, what are the physiological conditions where we see a constriction of the efferent arterial? I'm not sure specifically, but nonetheless, that is what happens when the efferent arterial constricts. So it's really kind of like the opposite effect on GFR as what we saw with afferent. All right. Okay. So I want to finish stuff off here, talking, kind of getting back into more details about the tubuloglomerular feedback, how that works. More of a visual as opposed to just writing. All right. So when we talk about the anatomy, of course, you guys know all about the tubules, right? The proximal tubule. We spoke about the capsule. We spoke about the glomerulus, afferent arterial, efferent arterial. This is a cross section through, and this could either be the distal tubule. This could either be part of the loop of Henle, but more likely it's the distal tubule. These dark cells, you notice it's mostly light, but these dark cells up against here, those are what are called the macula densa cells. Here's the afferent arterial, all right? As you can see right here, if there's an increase in blood pressure, increased GFR, this is going to translate into more solute passing into the tubules, passing through the proximal tubule, passing into the distal tubule. Once that increased amount of solute and fluid reaches the distal tubules, those macula densa cells say, whoa, there's too much here. Got to slow the kidney down. So it squirts out adenosine that triggers constriction of these afferent arterioles. So it's like right next door. So whereas the myogenic response, right, that's due to a direct effect, the blood pressure goes up through here, this constricts, right? With the tubuloglomerular feedback, that's after fluid has already passed through, it's detected in here and it squirts back to constrict the afferent arteriole. So in both cases, they respond to an elevated blood pressure, blood volume, which it leads to a high GFR. This is a response to help to bring it back to normal. And finally, this is really just an overview. This is basically the same thing. Here is blood being filtered. There's number 1, GFR, goes to the proximal tubule, the loop of Henle, goes to the distal tubule. There's the macula densa. And what happens is when that increased fluid, increased solutes, reach the macula densa squirts out adenosine, causing this to constrict. That's number 5. And thereby that's going to reduce the pressure going in and thereby help to maintain GFR. So that is it. Let's see, and stop share. All right, and so that takes us to the end of renal 2. Tomorrow we'll talk about renal 3. Any questions? Okay, I will see you guys tomorrow. Have a good rest of the day.